Thank you so much for the kind introduction. And I just want to congratulate Anna for putting together such a fabulous day. And thank you so much for inviting me to be part of this. And I'm delighted to start this panel. And I've been tasked to actually speak for the next 10 minutes or so on my research, covering the topics of brain, insulin, and metabolism. So it's a daunting task, but I'm just gonna, give you a little bit of highlight and give you some flavor for the kinds of research that we do in the lab. And, given that we're tasked to talk about brain, insulin, and metabolism, I'm gonna start with insulin. And just not too long ago, we had unfortunately, during the pandemic, had a very nice celebration of the discovery of insulin. And, less than a year later, actually, this product that was discovered right here in the University of Toronto went to a human, right? So at the Toronto General Hospital, that's a photo of Leonard Thompson, who received the first dose of insulin right at Toronto General Hospital, where I started my training, and full circle, I'm now a full professor there. And this is just an illustration that this is how I was introduced to insulin in the clinical setting. And it's just an illustration of what a powerful anabolic hormone insulin is. And that without insulin, there is this unrelenting catabolism that typically succumbs to death. So it's really a powerful hormone, which is highly pleiotropic. In the context of metabolism, we all know insulin to lower glucose levels, and it's regulated by a lot of counter-regulatory hormones, including glucagon. But really, insulin acts in every tissue in the context of metabolism. It suppresses lipolysis in the adipose tissue. It promotes glucose uptake in the muscle. It even acts on the pancreatic beta cells themselves and promoting beta cell function. And in fact, insulin also acts in the brain in the context of this panel discussion. Insulin crosses the blood-brain barrier, and indeed, insulin receptor actually exists in every single cell of our body. So, I mean, how daunting is this? And this is just in the context of, let's say, a neuron where the insulin action takes place. And this is probably a very simplified diagram of what happens molecularly. So, it controls anything from, disrupting the blood-brain barrier to amyloid deposition to inflammation. So it's just very, very daunting to really think about the actions of insulin. And now, we have to also think about insulin resistance, as Professor Phillips has talked about. And in our lab, we've been interested in this protein molecule called PTEN, which is one of the powerful endogenous negative regulators of insulin signaling pathway. And this is another sort of more simplified diagram of the PI3 kinase signaling pathway, which is one of the main branches of insulin signaling pathway, of which PTEN is, again, one of the key negative regulators, such that if you were to disrupt PTEN, this would then allow for enhanced insulin action. So with that, some 20 plus years ago, when I began my independent research, I wanted to look at the role of PTEN specifically in beta cells in the pancreas and alpha cells in the pancreas. And this was a natural place for me to go as an endocrinologist, having trained in signaling pathways. And what was very interesting is that even though PTEN in some context functions as a tumor suppressor, in the context of specialized cells like endocrine cells and probably also in the neurons, PTEN, when it's deleted and PI3 kinase is therefore allowed to be optimized and enhanced, this actually promotes protection against models of type 2 diabetes, also type 1 diabetes, but through very distinct mechanisms that I'm not going to get into. This is all published work from very talented graduate students. And I just want to highlight this paper, which was actually not the direction that I wanted to go to. But when we wanted to delete PTEN using the insulin receptor promoter, right? So we were using the Crelox-P system to knock out PTEN, because if you knock it out in the whole body, it would be embryonic lethal. So we always use a Crelox-P system, and you have to use a cell-specific promoter to delete a gene of interest. So in our case, we wanted to use the insulin promoter to delete PTEN in pancreatic beta cells. And what was very, very confusing at the time is that there was actually a lot of deletion in the brain. And when we dug the literature, it turns out that insulin promoter activity, at least, is actually produced active in neurons, right? Particularly during embryogenesis, but even in adults, there is insulin promoter activity. So, we scratch our heads, after med school and insulin using, I never thought about, insulin, not that insulin is the place, not that insulin is produced by the neurons. I mean, that's actually a topic of debate that I'm not going to get into today. But for sure, when we look at the fundamentals of genetics, right? And it turns out that insulin signaling pathway is highly conserved, going all the way down to the simpler, C. elegans. This is a very simple organism of, no more than 1,000 cells. And in these very simple organisms, insulin signaling pathway is highly conserved. But what's more important is that insulin in these simpler organisms is actually all produced by the neurons, right? And so we decided that the PTEN deletion in these insulin promoter expressing cells in the brain must have some significance. And we were particularly interested because these mice were highly, highly insulin sensitive, and they were completely protected from insulin resistance and diabetes and many, many more things that so we continued to work on this, and we stumbled on this phenomenon called anti-inflammatory reflex. And as any good grad student would do, they went to a talk. And we were fortunate to, in Toronto, we're just so fortunate to have all these superstars come into town to give talks. And in this particular time, we had Dr. Kevin Tracy, who's one of the, I would say, godfather of this phenomenon of anti-inflammatory reflex. So what he's been working on, he's a neurosurgeon by training. And what he worked for the last probably 3+ decades with very, very elegant papers and stories in Nature, Cell, and Science. And basically what he showed is that the vagal nerve, just like we have a motor sensory reflex, there is this reflex that's called the anti-inflammatory reflex that utilizes the vagal system. So basically how this works is that the afferent nerve of the vagus would sense the peripheral inflammatory state. And then the efferent would then mitigate and regulate. And this is how you would resolute inflammation. And without that, you would have this unchecked inflammation, which would often get into cytokine storms. And this is sort of the mechanisms of, full-blown inflammatory conditions like sepsis, or even these low-grade inflammatory conditions like, even depression that we talked about, and some autoimmune diseases like rheumatoid arthritis. And what's very interesting about the vagus nerve is that it does have a branch that runs relatively superficially at the neck. And now there are FDA-approved devices that could stimulate the vagal nerve, right? So this vagal nerve stimulation could then activate and dampen inflammation. And because so many diseases, including diabetes and insulin resistance, is very intertwined with low-grade inflammation, this is one of the areas that is very, very interesting and has a lot of potential. So instead of using the insulin promoter to delete PTEN, we decided to really focus on the brain. And we used this promoter called FOX2B. So this FOX2B promoter is exclusively expressed only in the cholinergic parasympathetic neurons. And my student using the reporter very nicely showed that it's only expressed in the dorsal motor vagal complex as well as the vagal nerve itself. So it's a very clean system now because we have to somehow separate from the pancreas, right? And only look at the vagal system. So using this system, and what we did was we wanted to take an unbiased approach and really go straight to the single nuclear sequencing. So my student, Andy Lee, what he did was he had to pull something like 18 mice to isolate the vagal nerve to get this data. And this is really powerful data where he, first of all, showed that even as what we would think as homogeneous tissue is as a nerve has many, many different cell types, right? So there's fibroblasts. I mean, neurons really constitute a very minor portion of the vagal neuron. Right? There's astrocytes and microglia. And on the right panel, there's a really nice proof that PTEN is only deleted in the neuronal population, the neuronal cluster, and it didn't really touch any other cells. So we're talking something like 5,000 cells where PTEN's deleted out of, I don't know, 9 billion cells in the mouse, right? So this is a very, very sort of rigorous way to test the role of PTEN and insulin signaling in the context of a whole living animal. And when we did the differential gene expression analysis, what was quite astounding is that there were many, many genes that were upregulated in the knockouts on the right panel. And what was quite interesting is that many, many of these genes are genes that are involved. First of all, I've never heard of these genes, but what's very interesting is that they're genes that are involved in neuronal growth and neuronal differentiation. And look at that, even, conversion of dopamine to norepinephrine. So lots and lots of different genes that are relevant for neuronal and nerve function. So what would that translate to? So we now took more mice and would do a cross-section of the vagal nerve, and you could see that these nerves look very, very different than the wild-type mice. This is after 3 months of high-fat diet, and you could see that these nerves are very highly myelinated. They have a lot of neurofilaments and all these anatomical changes that would be conducive to higher conductivity and perhaps better function of the vagal nerve. So then we collaborated with bioengineering at U of T, and we decided to directly test the vagal nerve. So we know vagal nerve is one of the major branches of the parasympathetic nervous system, and we know the function of it to cause bradycardia. This is one of the gold standards tests. And what we show here is that the more stimulus you need to cause bradycardia would mean that it has lower function. Whereas if they have higher endogenous function, you would need lower threshold. So you could see here that the knockouts, they have better vagal function such that they need less threshold for it to cause bradycardia. Now, what happens when you feed them high-fat diet? So the vagal function actually worsens. And this is actually true even in humans. So I had a visiting professor from Samsung University, and they have treasure trove of data. And she knew we were doing these experiments. And so she went back to her institute and decided to mine the data, looking at the heart rate variability, which is another readout of vagal nerve function. And what she showed is that poor vagal nerve function could predict type 2 diabetes onset. And she's since then shown this in the context of many, many other diseases. So what happens in metabolism? So as an endocrinologist, these are sort of our go-to experiments. And these are mice, remember, just PTEN knocked out in the vagal nerve. That's it. The rest of the mice have all the normal constitutive cells. And these mice don't gain weight. They remain glucose tolerant. They remain glucose insulin sensitive. And the last panel is a photo of MRI of the whole body. And the white is all the fat that would accumulate with high-fat diet. And these mice, the knockout, they don't have any adiposity. So these are really very powerful model system to study. And given we're talking about mitochondria, it'd be remiss for me not to talk about this. And it has all the favorable functions in the liver, in the adipose tissue, in muscle. You could see that there is enhanced markers of oxidative phosphorylation and beta oxidation. So there's a lot of work to be done still, but these are just a very powerful connection of what neurons and the nerve could have so much systemic effects in all the tissues. So with that, I'll end and thank particularly Andy Lee, UJ Lee, who's done all the work that I showed you. And I'm looking very much forward to the panel discussion. Thank you.
I think we have time for some questions. So are there any questions in the audience to start off with? Well, I had a question actually, and I'm just so fascinated by this work. Sorry, I'm allowed to break the system here. So the P10 finding, can you say a little bit more about how this might translate to some kind of treatment or intervention for diabetes or people with other sort of aspects of insulin resistance?
Yeah, thank you. So the reason why we wanted to really hone in on just one cell type, and it's important because, as I mentioned, P10 knockout in the whole animal is lethal because P10 is such a critical gene. And depending on what tissues that P10 would be knocked out, many of these sort of innately proliferative tissues like breast or uterine, if you knock out P10, it would promote PI3 kinase pathway and it would promote tumors, right? So P10 is, after all, I think a second most common tumor suppressor. So that is really why, I took the trouble, my grad student took the trouble to really knock it out only in the 5,000 cells, right? And here we could really unveil the functional importance of P10. And so I cannot emphasize enough the importance of the tissue-specific effects of PI3 kinase pathway and P10. And, in fact, so I put in a huge grant actually that involves neurosurgeons and bioengineers to really translate this, right? And its effects in influence. So this is sort of just the beginning of the end and the beginning. So yeah. And in fact, we just got a grant with a neurosurgeon to look at this particular mouse in the context of cognition and depression. So we just got a grant for that. So there's a lot more work that we're interested in that I've been wanting to do this, for a decade or more. So, but stay tuned.
Thank you. I was going to ask about the cognition, but I think there's another question. Yes.
Mm-hmm.
You had a macrophage
Yeah.
sort of drawn and acetylcholine. So I'm just curious about how does this work?
Yes, so thank you for that. So, it's hard to cover such a complex topic in 10 minutes. Thank you, Anna. So, it's a very complex question, and there's lots of elegant work by Tak Mak and Kevin Tracy, where, what we learned in medical school about parasympathetic and sympathetic, it's not that simple, right? And even studies that are done even, 30 years ago, maybe not true, right? So we did think of insulin action being mediated through the vagus, but it gets a little bit complicated in that the parasympathetic then innervates so it's somehow then transferred to a sympathetic, okay? And then it's the sympathetic nerve that then innervates the spleen, okay? And then in the spleen, through lymphocytes, the lymphocytes are the source of acetylcholine, okay? And this also involves the myeloid cells. So I didn't have time to show the data, but when we deplete myeloid cells, then the phenotype goes away. So that's how I could say that this is through modulating the inflammation. So we are using these mechanistic approaches to really, reductionist approach to show that it is ultimately, at the end of the day, T cells and then myeloid cells that have the alpha-7 acetylcholine receptor. So we've done a knockout of that as well in our old paper. And that also abolished the phenotype. So we've done all these reductionist approach, knocking out each of the components of this pathway to allow me to say that it is involving the macrophages through the alpha-7 receptor and the acetylcholine activation to the cognate receptor in the macrophages that ultimately control the inflammation systemically.
I think we might have time for one more question. I'm not sure how to access the online, Anna, but yeah, okay. Can I just ask one question, maybe naive? The translational relevance, so sort of conservation across species. Can you say a little bit more about what happens in humans with the P10?
Right.
neuron
So that's a very good question as well. So first of all, I just find it fascinating that in the lower organisms, let's say the C. elegans, it's in my slide, but there's something like 37 insulin homologs, which I find, and in those lower organisms, insulin seems to do everything, right? Like reproduction and growth. So, and then I guess as the system got more complex, the pancreatic beta cells sort of took over the insulin. So in humans, there's only, INS1, INS2, and then IGF-1, IGF-2. That's it, right? So that part is interesting. In terms of getting back to your question about P10 in humans, so there is a syndrome called Cowden syndrome, where they have P10 mutation. And so this paper was in New England Journal by Anna Gloin, who's one of the geneticists, Oxford. Now she's at Stanford. And basically these collections, in fact, I have one of the patients actually that I look after because she had thyroid cancer. So Cowden syndrome, it's also called hamartoma syndrome. So they present with benign tumors, right? So including thyroid and breast. So my patient already had like bilateral mastectomy, sort of prophylactically, and she's undergoing surveillance. But she did have papillary thyroid cancer, so she had thyroidectomy. But what was in that New England paper is that these patients have high insulin sensitivity, right? So again, that's a very genetic sort of illustration of this conserved evolutionary function of P10.
And cognitive ability, obviously it's N equals 1. I'm just curious to know how it relates to brain function and cognition.
Yeah, no, that's a good question. So I have not brought this up with my patient, and I'm not sure but I should talk to Anna Gloin and see if she has the collection of patients and whether maybe, I don't know if in Toronto we might have enough cohort to do that, but I'll certainly talk to my colleague, geneticist, who's looking after her as well.
Well, thank you very, very much, Professor Wu. That was really fascinating. I could go on and on asking questions, but I think we have to move to the next speaker. So thank you. Let's give a big round of applause to Professor Wu.